3.
Hi friends, welcome back to my channel. Today I am going to start one important mechanism that is known as oxidative phosphorylation, belongs to biochemistry. So let us start. So here, what is oxidative phosphorylation? So oxidative phosphorylation or electron transport linked phosphorylation is defined as the metabolic pathway in which the nutrients undergo oxidation to produce the energy in the form of the high phosphate compounds, nothing but ATP molecules, are known as oxidative phosphorylations. The oxidative phosphorylation, that is the mechanism of oxidative phosphorylation, is explained by three theories. First number one is called as chemical coupling hypothesis. Chemical coupling hypothesis. Second one is known as chemiosmotic theory. Chemiosmotic theory. Third one is known as conformational coupling hypothesis. So these are the major theories which explain the oxidative phosphorylation. The most commonly accepted hypothesis is known as chemiosmotic hypothesis. So let us start the chemiosmotic hypothesis. So first we draw the mechanism, how it's explained. So everybody knows the mitochondria have the inner membranes, outer membrane and intramembrane space. So now we are drawing here. Suppose this is for matrix. So it made up of intramitochondrial membrane and intramembranous space. So now we are designated that is known as intermembranous space, and this is known as inner mitochondrial membrane. Inner mitochondrial membrane, that is called matrix. So now in the inner mitochondrial membranes, what is there? Hydrogen, potassium, hydrogen ATP molecules is there. We are drawing here, that is known as complex phi. And others electron transporters, that is complex 2. We can draw like this. Then complex 3 and complex 4 can discuss. Now, so here this is known as F1 F0 protein, there is F1 proteins. Now this F0 protein acts as a proton channel by which it can enter the proton inside, that is 10. Then here 10 hydrogen ion, then it translocated from the matrix to the intramembranous space to the complex 2, that is 4 hydrogen, and here 4 hydrogen, but in case of complex 5 here 2 hydrogen. So this is coming to this is called proton gradients. Here it will come, this is a proton circuit, here coming this one. So here will be pH gradient between this one, pH gradients. So here it will be OH minus and outside the number of the H plus ion. So here this it will be plus, inside will be negative. Inside will be negatives. So the intramembranous space, the pH will be 1.4. It is very less the outside that is rather than the matrix. So now So everybody knows the complex 2, here it is there. Complex 2 means nothing but the name of the complex 2, what is the name of the complex 2? Complex 2 is known as NADHCOQ reductase. Whereas the complex 3? Complex 3 is known as COQ cytochrome C reductase. Whereas complex 4 cytochrome oxidase. So now let us start how it is happening. So here, so everyone knows that mitochondria consist of inner membranes, outer membranes and intramembranous space. So here the mechanism of the oxidative phosphorylation is explained by chemoosmotic hypothesis. So what does it state? The chemoosmotic hypothesis states that how the transport of the electrons effectively utilized to form the ATP molecules by combining the inorganic phosphate, that is H3PO4, that is phosphoric acid, along with the ADP with the help of complex 5, that is known as ATPase or hydrogen ATPase. So here the chemoosmotic hypothesis is comparable with the energy stored battery, which is separated by positive and negative charges. Now the chemoosmotic hypothesis state that, or it indicate that here the mitochondrial inner membranes is the prerequisite for the oxidative phosphorylation. Now what is the exact mechanism? So here generally the inner mitochondrial membrane are not permeable the hydrogen ion and hydroxyl ion. So due to the proton gradients. So here we told here F0 and F1. So F0 and F1, this is a complex 5, means F0 it is attached with the inner mitochondrial membrane, whereas the F1 protein it is projected inside the matrix. The F0, the pores of the F0 act as a proton channel, means it translocates the protons or hydrogen ion from the intramembranous space or inner mitochondrial membrane, that is known as intermembrane space, to the matrix. So now when the hydrogen ion pump from the matrix to intramembranous space, so what will happen? The pumping of the hydrogen ion from the matrix to the intramembranous space with the help of the complex 2, complex 3 and complex 4, it develop a potential difference between the intermembrane space and matrix. What will happen when the hydrogen ion pump from the matrix to the intermembrane space? There will be a decrease the hydrogen ion concentration, but increase the hydroxyl ion. Increase the hydroxyl ion. So what will happen? The pH of the matrix will be rises. That is why it exists in the OH minus, means the number of the hydroxyl group is increased. So inside of the matrix is negative, and whereas the intermembrane space are accumulated by a more number of the hydrogen ion, so it decreases the pH, that is 1.4. so this is existing the positive charge. So means the inside of the matrix pH is high, whereas the intermembrane space pH is low, so it generates a potential difference, means pH gradients. So these pH gradients are known as proton motive force. These proton motive force dehydrate the ADP plus inorganic phosphate and drive it to form the ATP molecules, that is here molecules 2.5 form the 2.5 of the ATP molecules. So the proton motive force drive the hydrogen ion from the inner membrane to the matrix, and it leads to the combination of the ATP molecules with the inorganic phosphate to form the ATP molecules. So due to this, each ATP molecule formation lead to the pumping of the proton from the matrix to the intermembrane space with the complex 2 is 4 hydrogen ion will be pumped from the matrix to the intermembrane space through the complex 3, it will give also 4 hydrogen ion, and but in case of through the complex 4, it will only form the 2 hydrogen ion. So in total, the formation of the ATP molecules, 10 hydrogen ion must be pumped from the matrix to the intramembrane space. So this is the major mechanism for the oxidative phosphorylation. Now different factors or chemical inhibit the oxidative phosphorylation. So what will happen here? Complex 2, what are the factors or what are the chemicals which inhibit the oxidative phosphorylation? So here first, the complex 2 means that is known as site 1. So site 1 is inhibited by the chemical that is known as rutinone. Rutinone is a fish poison which inhibits the transfer of electron from the Q NAD to Q reductase to ubiquinone. So one is called rutinone, then another is called amobarbital, sacobarbital, and drugs chlorpromazine, they can inhibit the site 1, that is they inhibit the complex 2. In the complex 3 means site 2, it is inhibited by the antimycin A, British antiluticide or dimercaprol, and another is called fenformin, that is oral hypoglycemic agent, that is anti-diabetic drugs. So drugs that is called fenformin to inhibit the electron transfer in the complex 3, that is site 2. Next one, complex 4, that is cytochrome oxidase, is inhibited by hydrogen sulfide, carbon monoxide, cyanide, and azide. So these are the inhibitors for the electron transport chain. But here another thing is there, metabolic uncoupler. What does it mean? Metabolic uncoupler means here the F0 allow the proton from the inner membrane, intermembrane space to the matrix. So it drives the formation of the ATP. But along with this complex, other protein are present, which also transfer the proton from the intermembrane space. But here there is no formation of the ATP, as because in that protein the ATPs are absent. ATPs are absent. But phosphorylation will be takes place. But when that is called UPC, that is called uncoupler protein. UPC. This is called UCP. Uncoupler proteins. It is also present in the inner mitochondrial membrane, which also responsible for the transfer of the proton from the intermembrane space to matrix. So it will lead to the phosphorylation, but highest concentration of the oxygen is consumed by the matrix. So what will happen? It dissociates the oxidation. But in that case, the energy will not be released in the form of the ATP molecules, as because in that UCP there is no ATPase enzyme. But oxidative phosphorylation will be happen. But in that case, the energy will be released in the form of the heat. So this is known as metabolic uncoupler. The metabolic uncoupler example means the chemical substance which increase the proton transfer from the intermembrane space to the matrix. So example 2,4-D and P, that is known as 2,4-dinitrophenol. Second example is called CCP, means chlorocarbonyl cyanide phenyl hydrazone. Chlorocarbonyl cyanide phenyl hydrazone. And that is known as FCC, that is called trifluorocarbonyl cyanide phenyl hydrazone. So these are the chemical metabolic uncoupler which increase or permit the hydrogen ion from the intermembrane space to matrix, but they dissociate the oxidation. The energy will be released in the form of the heat, not in the form of the ATP molecules. Next, the physiological uncoupler, thyroxine and thermogenin, both are the physiological uncoupler. So next, these are the examples for the metabolic uncoupler. So here, so in I told that these mechanisms are responsible for the production of the energy, that is mainly ATP molecules. Now some deficiency is there. For example, cytochrome oxidase. If the cytochrome oxidase is nothing but the complex 4, if there will be a deficiency in the cytochrome oxidase, what will happen? The one disease is developed, that is called malaise. That is called mitochondrial encephalopathy, lactic acidosis and stroke. It is defined as the inherited disorder characterized by the deficiency of the complex 4. It will lead to the mitochondrial encephalopathy, lactic acidosis and stroke. Next, here last one theory is called conformational coupling hypothesis. The conformational coupling hypothesis, it is explained that the F0 and F1. So F0 is what? Fernandez-Moran proteins, and F1 is the Fernandez-Moran one. Why it is called F0? Because this protein is inhibited by the oligomycin. So here the inhibitors of the electron transport chain I explained here, but metabolic coupler also I explained. But what is the inhibitors for the oxidative phosphorylation? There are 3 inhibitors. One is called oligomycin. Inhibit the transfer of the proton from the intermembrane space to matrix. Second one is called atractyloside, which inhibit the transfer of ADP to ATP or ATP to ADP from the matrix to cytoplasm or cytoplasm to matrix. Third inhibitor is called Bongri gate, which also inhibit the transfer of the ATP from the matrix to cytoplasm. So these are the 3 inhibitors for the oxidative phosphorylation. One is called oligomycin, second one is called atractyloside, third one is known as Bongri gate. So these are the 3 inhibitors for the oxidative phosphorylations. So the mechanism is up to this. Thank you all my dear friends. If you like my channel, please subscribe. Thank you.